Hi everyone, my name's Alyce. I'm a postdoc at the Wellcome Institute, and I'm going to give a fairly broad overview to comparative genomics. I'm going to take you through a fairly generic pipeline or several pipelines to explain what to do once you've sequenced a genome. As I said, I'm a postdoc. I'm working on cholera genomic epidemiology. And I did my PhD in chlamydial genomes and metagenomes. So two fairly interesting, from my perspective at least, bacterial species. So you've sequenced a genome. Now what? You essentially now want to use this genome to actually answer some biological or epidemiological questions. So the first steps, aside from some quality control, is to assemble and annotate the genome. And this is going from the process of going from reads to contigs to genes and other features. And I'll go through this in detail shortly. I've got a lovely flowchart coming up. And when I say investigate the genome, you can investigate several things. You might want to look at the sequence or the genetic structure in terms of the gene arrangement. But then often what you're really looking at when you look at those things is the function and how the sequence relates to the function. And then overall, you want to compare your new genome or genomes with other genomes. And this could be in several different contexts, and I'll touch on that shortly as well. And again, you might want to be comparing the sequence or the function. On the right here, I've got a few really commonly used tools, and I'll touch on most of these. Some of these are command line tools, which require you to have a little bit of coding experience. You use the terminal and type scripts in, or rather you might learn to do that as you go, as I did in my PhD, and I know a lot of other people had the same kind of pathway. But some of these tools also have graphical programs that have a much less steep learning curve, and some of these are really useful for visualization as well. But all of them are really commonly and frequently used tools in the microbial genomics space. So the final desired result of these analyses is what I call pretty pictures. And what this really is a clear way to communicate the results of your analysis, either with colleagues in a meeting or more formally in a publication. I'm a very visual person, and I know that a lot of other scientists are as well. And so if there's a warning here, there's lots of schematics and flowcharts through this talk. But I really do believe that a picture speaks 1,000 words. You can really convey a lot of information in a single picture in a slide or a publication or something like that. So one that we've got here, for example, is a really common one where you have what's called a phylogenetic tree, and this is showing the genetic relatedness between genomes, and you can overlay onto this tree any metadata or genetic or phenotypic information, and this is going to really help you communicate what you've found in your novel genomes and how your novel genomes compare to other genomes. But why might we do this in the first place? So the first and foremost reason, and the most kind of applicable one to you, is for outbreak investigation. So you might sequence one or several outbreak strains, and you might want to compare these to each other and see how related they are, maybe make a transmission network and identify transmission chains. Or you might want to compare this outbreak to previous outbreaks or to endemic strains. You can do all of these with genomics. You might also want to look for niche or host-specific functions and adaptations. You might want to compare asymptomatic cases with disease cases, for example. You can look at things like the virulence profile, or you might have other groups that you might want to compare, such as environmental or clinical isolates. And then you can also do things like antimicrobial resistance monitoring in the environment or, again, in clinical cases. And genomics is a really good way to complement lab-based assays and phenotypes. So if you've seen a really interesting phenotype in the lab, you can then search the genome for explanations of these phenotypes. So I promised you some flowcharts. This is the first one, it's the main one, and I realize that it is, there's a lot on here, it can be a bit overwhelming. I will go through it in general now, and I'll go through each step in more detail on individual slides. So we've sequenced our genome, we've got what's called our reads, and they're the starting point for this pipeline, and I'll show you what a read is shortly. We can then go down, so we'll go down this de novo approach first, and so this doesn't make any assumptions basically about what we've just sequenced. So we do first what's called de novo genome assembly, and that means that we're assembling our reads into what's called contigs, which is a contiguous sequence. Hopefully we assemble a whole genome. There's some quality control checks along the way, either at the read level, assembly level, and throughout the pipeline. I won't go into detail about those, but they are good just to keep in your mind. Once we've assembled our genome, we do what's called annotating the genome, and that is the process of adding features, so genes, and also things like ribosomal RNAs, transfer RNAs, adding these onto the DNA sequence. And what this will allow us to do is to go down sort of a preliminary investigation pathway where we can start to characterize our genome, or we can take the next step and run some comparative genomics pipelines. And I'll talk a lot more about Pen Genome analysis shortly. But one of the really cool things that this gives us is a core genome alignment. And this is something that we can use as input for the phylogenetic tree, like I just showed. And this really is the basis of a lot of our pretty pictures. We can add in the metadata that we've got for our samples, or any other phenotypic data, as I just mentioned. So this contrasts with this other pipeline in grey, which is a reference mapping pipeline. We essentially get the same result of phylogenetic tree, but it's made a little bit differently. So this pipeline makes the assumption that we know what we've sequenced and that it's close enough to an existing genome that we can use that as a reference and do what's called variant calling. So we can call variants of our novel that we see in our novel genomes relative to this reference. And what we get after that is what's called a Snip alignment, so a single nucleotide polymorphism alignment, where we're only taking the bases that are the positions that are different across our input genomes, and that might be 2 or it might be 200. Instead of using a core gene alignment as input for our phylogenetic tree, we use this Snip alignment. And again, I'll go into both of these in more detail as we go on. So on the right, we have what's called a read file. And this is called a Fastq file, and the Q stands for quality. For every sample, we have 2 read files, a forward and reverse, and these are what we call paired end reads. So for each sample, for each direction, we have a lot of reads per direction, and each read has 4 lines. So we can see here we've got the line starting with @ and that's our header file, our header line. And so that will have sample and lane information, and it has which pair of the reads we've got. The second line is the actual sequence, so a series of G's, C's, T's, and A's. Our third line has the plus symbol, so we're adding the sequence and the quality together. So the final 4th line is the quality line. And this has, this looks a bit messy. It has what's called Fred scores, and that's from 1 to 40. But this is quite hard to put in. You can't put 1 to 40 in these lines to match the individual bases. So instead, what's used is letters, numbers, and characters. So we've got A to Z is, so that the best quality is A, and then we've got A to Z, and then numbers, and then characters. So you can see here, I've got the direction here, and I've got a really high quality A. At this position, we've got a quality score of A or 1, and then we've got a really low quality score. Here, the base caller actually couldn't tell if it was an A, T, G, or C, so it's called an N and assigned a low score. We have some quality control steps that we can do at this stage, and one of them is called Fastq, where obviously it's not very efficient to open every read file for every sample and look at it like this. It's not very quantitative, so Fastq and another program called MultiQC will take all of your Fastq files and essentially summarize the quality scores for you. And from then you can either choose to keep all your reads, keep some of the reads, filter or trim some of the reads depending on their quality. It's really up to you depending on what your quality is like to begin with. The second tool I've got called Kraken is something that will assign your reads to taxons based on databases. So this is really helpful if you don't particularly know what you've sequenced. This will be the first kind of taxonomic identification, or if you suspect there's contamination, or you want to rule that out, this is the first indication that you've got that. So once you've got your reads and you have checked their quality, you can start the de novo genome assembly. And to do this, we use a tool called Spades. This is really the main part of the process. Everything else that we do after this really relies on this. And what de novo actually means is from scratch. So we're taking the reads and we're assembling them from scratch. And I'll show you the process of this. So say, for example, we've got our read set here. This is just one sample. And we've got our forward and reverse reads in blue and red. These aren't actually meant to be the reverse complement of each other. This is what I first thought when I started out, but this isn't actually the case. It means that there is what's called an insert of a known length between the forward and reverse. So we don't know the sequence, but we know how long it is. And again, this is another indicator of sequencing and read quality. So we can see that there's light and dark shaded reads, and I've done this on purpose. So if we take our light shaded reads to begin with, we just separate them out. If we essentially align them all together, I'm missing some complicated maths, but I just want to show you a schematic, sort of way of what this is actually doing. We can see that the reads overlap at all of the positions, and our forward and reverse start to overlap as well, which is exactly what we want. So we have what's called contiguous sequence. So we assemble these into our first contig. So I've got a nice long contig here. Let's then take our dark colored or dark shaded reads, again forward and reverse. We'll do the same thing, we'll align them to each other. And what you can see is that there is overlap, but we do actually have a little gap here, a little break. And so what the assembler will do is that it doesn't know what to do in this region, so it will make, instead of one contig as we did with the light shaded ones, we've got two contigs. So then we've got our two dark purple contigs and our one light purple contigs. So these are our three contigs. For the purpose of this, we've just assembled a really lovely genome with one chromosome and two plasmids. This almost never happens. I just wanted to make a nice, neat example. So what does a contig actually look like? Again, we've got a text-readable file with, again, a lot of C's, A's, T's, and G's. We've got our header sequence, which is a little bit different to the Fastq file. Instead of having an @ symbol, it's got the greater than symbol. We've got our sample name after that. And we might have one long contig and then a series of smaller contigs. So each contig has their own header, as you can see here. And we call this a Fast A sequence. or faster instead of a Fastq. And like I said, this is the main input for a lot of the downstream analyses. So once we've assembled our genome from scratch, from our reads, we can then do what's called genome annotation. And we use a tool called PROCA to do this. It stands for Prokaryotic Annotation. And once we've got this information, we can start to do some investigations. We can start to characterize the genome, become familiar with the genetic structure, and we can use a tool called Artemis to do this. And I'll show you that in a moment. But first, the output of annotation. So this is called a Gff file. And what you have is a list of your contigs. You have your Fastq sequence, so the actual DNA sequence, and then underneath that you have what's called your features. So each contig has then a list of features annotated to it. You have things like coding sequences and tRNAs. You have a product name, so this particular one is a nitrite response regulator. And then each feature also has what's called a locus tag, which is a unique identifier. So I'll show you all of these in a slightly nicer way. So once again, a text file is not very useful for investigating a genome. So we can open the Gff file, which is the genome feature format file, in a program called Artemis that I just mentioned. And I can see that this is very overwhelming at first. I did not like it at all when I first used it, but it is quite intuitive once you get to know what the panels are and how to navigate around it. And you can really then start to get a feel for the genome structure, the distribution of genes, operons, genomic islands, that kind of thing. So I'll talk you through this, and I'll show you a few of the features that can help you start to investigate and characterize your genome. So the top panel is the strand and frame nucleotide view. So these blue things, these blue vertical lines, are all coding sequences. And so we can see that there's 3 lines above, and then we've got 2 central lines, and we've got 3 lines below. So what these are is the central lines are the DNA strands, forward and reverse, or reverse complement, I should say. And then we've got our 3 forward frames and our 3 reverse frames. The middle panel is the exact same thing, but generally the amino acid view. So you still have your 2 DNA strands here, and then you have the amino acid translations, again in all 3 frames. And you can see again that there's mainly letters here, so for the amino acids, and then we also have these symbols, and that's for the 3 different stop codons. This panel is best zoomed in like this. And then the final bottom panel has all of our annotation information. So we have what's called the locus tag, which is the unique identifier of every feature. We have the start and end position of each of the features. In this case, it's mostly coding sequences, but our contig, or in this case, a chromosome, also is also a feature and also has start and end positions. And then we have the C is for which strand it's on, and then we've got the name of the product of the gene. So a little less overwhelming once you actually know what you're looking at. So if we click on a coding sequence, so I clicked on this first one here, you can see that it has now, the middle panel has now moved to the start of that gene. And you can do a few things here. You can modify this gene, you can extend it or shorten it depending on how well it's been annotated. Sometimes you need to extend the start back to the previous start codon, for example. The annotation, as amazing and useful as it is, it's not always 100%. So sometimes you have to do some things manually, and I will show you an example of that shortly. And then we start to zoom in a little bit more and start to get a bit more clarity about what we're actually looking at in the top panel. So we start to see some black lines here, and what they are representing are stop codons. So you can see that there's a bunch of stop codons mixed in with these grey sections, which is everything else. And then we have these regions that are shaded in blue, which are our coding sequences, and they're between a start and stop codon essentially. So it's an empty reading, an open reading frame, an ORF, and then when so Proca, the annotation tool, it has a couple of different steps. It first looks for empty open reading frames, and then it searches within each of those open reading frames against databases to look for sequence similarity with coding sequences. And then it makes its best prediction about which ones are real and what they actually are coding for. So you can see, zoomed in one more time, you can see that the bacterial genome, and I think I should also actually say this is a Vibrio cholerae genome. This is the reference genome that a lot of people use, N16961. So I am showing you a relevant example. You can see that the genome is very tightly packed. And when I say that, I mean that there are a lot of coding sequences, one after the other, without very much space in between. You do see, obviously, some bits of space. And there's a little bit of overlap, but you generally won't see very much overlap, and you won't see overlapping genes on different strands in general. Sometimes, as I've just said, sometimes these are slightly misannotated. You might have things that don't span the full empty open reading frame, or they might be missed completely. So if we click on a coding sequence, it is highlighted red, and it brings us down to, in that middle panel, it shows us the amino acid view of that coding sequence, and then it also takes us to that gene in the bottom panel as well, so we can get a bit more information. That way, no one's going to know exactly what VC2773 is. I mean, some people might, but I don't, so it can take us to that information. And if we then view the information about the feature, I'm in the edit function at the moment, we can start to see this same information that I showed in the Gff file, just in the text version. All of that same information is here. So this tool has read all of that information and essentially translated it into these graphics, and we can still pull out all of that same information. Again, I'm in edit, so you can modify the gene, the feature here if you need to. So just as an example, I'm going to change the color from the very garish bright blue to the slightly nicer darker blue. That's encoded by the color 4 in this case. Might not seem very helpful, but it's good for demonstration. And occasionally it is actually helpful. I've used this feature before. If I want to color all the genes in a particular operon example, for example, and then I can select all of those genes and extract them into a single Fastq file. It's quite an easy way to do that. So putting some of this together now, I found an empty open reading frame. That's this one just here. And I didn't make this, by the way. This I didn't just sort of do it for the example, but it wasn't annotated properly. And it's conveniently located near some genes that have been colored white by the people that submitted this genome. And this also happens to be the CTX phage. So I can see that there's probably an empty open reading frame here that probably encodes for a real gene. So if we want to make some changes to this and we want to investigate what it actually might be, we can select this region. And as you can see, it selects in all the panels. So it selects, I've selected here in pink, it selects it on the DNA strand as well, and it selects it below in the amino acid view. We then can go and create a feature from this base range, and I'm going to change the color as I did just before. I'm going to make it pink so that it stands out a little bit. And because I don't know what it is yet, I'm going to designate the product as a hypothetical protein. Once we've then made this coding sequence, we can do the modification as I mentioned before. So first we have to make sure that it ends on a stop codon, which I somehow managed to do perfectly. That's the hash symbol. And then if we go to the other end, I've extended this to the first methionine after the most recent stop codon before that. So we've created a feature, we've given it a preliminary identification, and we've made sure that it starts and ends properly. What we then need to do is try and figure out what it actually is, or at least what it has similarity to in the databases. So if you go up to run, there's a whole range of tools that you can run from within Artemis. And one of the ones that I use quite a lot is an Ncbi Search. And there's a couple of different ones here. So Ncbi being one of the large databases, and we can run a range of BLAST algorithms with our coding sequence. In this case, I'm going to run a BLAST-N, which is a nucleotide BLAST, and I'll also do a BLAST-P, which is protein BLAST. So what this does is it opens up a web server, and it gives us first an overview of what we've just submitted. So we've submitted a coding sequence, and it's given us the coordinates and which orientation it's in, which database we're running the BLAST against, and then also telling us the molecule type and the query length, which this is kind of a quite average gene size, so it's very possible that it's real. And so if we scroll down, we can see, so we've got all of our what's called hits here. So luckily they're all hits against Vibrio cholerae. They are all high identity and high quality hits based on these two 100% columns, and this E value, which is like a probability score. Probability of it being random is 0. So we can be pretty confident in these hits. That said, we're not actually getting a whole lot of information from this, because one of the downsides of many genomes currently being available, and this just always increasing, is that if you are searching for something that's reasonably well studied, you're not going to get a gene ID necessarily from a nucleotide BLAST. And that's one of the reasons that we'll run a protein BLAST in addition. So we do get some phage sequence hits, so we are getting a bit closer, but in this particular instance where we want to modify the annotation, we want to actually know what gene it might be. Just before I go to the BLAST hits, I do just want to show one more thing. So this is what's called an alignment. This is I'll show a couple of different ways that you can view alignments, but this is quite an elegant way, I think. You have your query, which is the sequence that we just found in our genome, and we have the subject, which is what it's matching against in the database. And so you can see there's a line for each sequence, and you can see that they're all the same. All the bases are identical based on A, the fact that you can see the bases, and B, this vertical line between all of them. So if we had a mismatch, we wouldn't have that line, and you would see that, for example, this A might be a C. So again, we have the same thing for protein BLAST, but it's telling us that it's an amino acid sequence, and that our query length is much shorter, so 1080 I think it was, divided by 3. So that makes sense, luckily. And then we go to the hits, and you can see that we've got quite a different range of hits here. We've got far more specific hits, and we're getting matches against the replication initiation factor domain containing protein, aka RSTA. And again, we check our identity and our quality scores, and we've got quite high quality and high identity hits here. So we can be quite certain that A, this empty open reading frame actually contains a coding sequence, and B, we can be fairly sure that it encodes this OSTA. So then we can go back to Artemis and we can update our annotation. So I will change the product to the one that we just found. I'm not going to read it out again. And we can also add, so these are called qualifiers, by the way, all of the information about a particular feature. So we can add a gene qualifier as well, and I'm going to make the RSTA. And so you can see over on the far left, some of them have gene names, and if a gene name hasn't been designated, we've got the unique locus tags. What I should have also done, and it's too late now, but I should have also given this a unique locus tag as well. In this case, I would probably make it, I'd have to, fit it in before 1465. I might make it 1464B, and then make RSTB to 1464A, just so that they were both unique. So if we go back to the flowchart for a moment, we have sequenced our genome, we have assembled the genome, and we've carried out appropriate quality checks along the way. We've annotated the genome and we've done some preliminary investigations. Most of what I've talked about so far in this pipeline has been from the standpoint of sequencing and characterizing a single genome. I mean, hopefully in doing this, I've helped you understand the process of going from reads to a genome. And this applying this to a single genome is really helpful for basic biology and for full characterization of a novel isolate. You can relate it to a phenotype, for example. And the tools I've put over in this box here are really good ways to do this. There's an absolute plethora of ways that you can investigate and characterize a novel genome. I mentioned right at the start, AMR and virulence profiles are really common things to look at, for example. However, the next step is to compare some genomes, either to compare your genomes to previously published genomes, or to you might sequence a set of genomes at once. So you can use a lot of these same tools as well as other approaches, and usually in conjunction with a phylogenetic tree, as we're working towards in this pipeline, in order to compare some genomes. And this is what we refer to as comparative genomics. With the final output, as I've shown again at the start, is you have some pretty pictures, often with something like a phylogenetic tree, and then some metadata and genotypic or phenotypic data alongside it. So one of these approaches is a Pen Genome analysis, and I've already touched on it, but I'm going to go into a bit more detail about it now. And this is one of the things that you do with your annotated genomes. This is really handy because we get multiple useful outputs. So we get what's called a core gene alignment, as I've already mentioned, and I'll go into detail about this now. And this is what we use as input for making a phylogenetic tree. We also get a gene presence-absence matrix, and as the name suggests, it's literally a binary matrix where you have all of the genes in a pan genome, and whether they're absent or present in all of the genomes that you're comparing. This is really good because you can query for target genes or operons, or you can compare presence or absence of genes across various groupings, either that you see in a tree or other groups from your metadata, such as asymptomatic versus disease or anything like that. And finally, we get some statistics outputs as well, which will tell us a bit about the evolution of the species or the strain or whatever level that we're comparing. And again, from all of these, you can get some pretty pictures. So I'll go into a bit more detail now about what a pangenome is, how we get this core gene alignment, and some of the pretty pictures that you can make, because Florent will talk about this later as well. So first up, just to define a pen genome. So I'm going to run through an example taking 3 genomes. We've got the purple, the green, and the blue genomes. And for this example, they are the same species, but they belong to 3 different lineages. And maybe those lineages have different ecological niches. But for this example, they're a similar genome size, and they have the same number of genes, so 4 genes each. They're quite small. So if we put these 3 genomes together, we can see that there is some overlap of all 3 genomes. We can see that there's overlap by 2 at a time for each combination, and then there's a region of each genome that's unique to that genome. So if we take first the first 2 genes in each genome, they are the same between the purple, green, and blue genes. They're not the same as each other, but from each genome, they're homologous. So they would be in this middle section, overlapping by all 3 genomes, or shared between the 3 genomes. If we then take the 4th gene, it's shared only by the green and blue genomes, so it would belong in this overlapping section, and that would leave the purple gene at the same position to be unique just to the purple genome. And then finally, this 3rd gene in each genome is completely unique to each one. It's not shared at all. There's no homology. So they would each again belong in these individual strain-specific clouds over on the side. So where we have the genes that are shared by all 3 genomes, they're what's called the core genes. And we're going to colour them in red for the rest of this demonstration. We have the single gene that is shared by both the green and blue genomes. That's the start of the accessory gene set. So anything that's not core is accessory. But even though they're shared by 2 genomes, they're still accessory. You can essentially have different levels of accessory genes, essentially. I'll touch more on about that shortly. Then we have the remaining genes that were not shared by any of the genomes, so they were completely unique or strain-specific to each genome, and they're part of the accessory genome as well. But as I was just mentioning, it's quite helpful. Again, we're talking about communicating these results and finding patterns. It's quite helpful to sometimes speak of these separately as being strain-specific or maybe rare genes, rather than just the blanket accessory genes. But just to demonstrate that a little bit further, I'm going to colour the accessory genes here all as yellow. So we can see that in our 3 genomes and our 4 genes, we have half of the genome is core and half of the genome is accessory. And that accessory genome is a little bit different across each genome, but that's okay. And that's what we'd somewhat expect, depending, of course, on our data set, how diverse the data set is that we're comparing. So essentially, the core and accessory genes together make the pen genome, but more specifically, the pen genome is all of the unique genes. So we started with 4 genes per genome, so we would have a maximum of 12 genes in the pen genome of these 3 strains. But we already know that some of these are shared by 2 or more genomes. So once we start removing redundancy, we start to get the total unique genes, i.e. the pen genome. So first, if we remove the redundancy in the core gene set, we're left with just one representative of each of those orthologous genes. And that would just be, say we would take the gene from the purple genome as the representative of that orthologous group. So that's the core gene set. This is usually referring to 99, in this case I've used 100%, but it often refers to the threshold of 99%. So if a gene is conserved in 99 or more % of your data set, this is set by the user. So depending on your data set, you might actually need to change this. You might bring it down, you might keep it at 99%. And that really, you do need to know a little bit about the diversity of your data set. Or you run it in complete default mode, and then you run it again after you've got your first results, and you temper the threshold based on the results. And so we've got 2 genes in the core genome there. Then if we remove the redundant accessory protein gene, remember that the purple and the green and blue gene at the 4th position was the same. So again, we only need 1 representative of that one. That leaves only the unique accessory genes left, and we've got 5 of them, so 3 and 2. And then within that accessory set, I've recolored the unique genes now, so you can more clearly see which ones are unique. Again, coming back to the idea that sometimes it makes sense to have subdivisions in your accessory genes, gene sets. We can hopefully see that we've got 7, with the addition of these 4 strain-specific genes, we have 7 total unique genes, i.e. the size of our Pen genome from these 3 small genomes is 7 genes. To look at this in a slightly different way and show you a little bit more about what you can do with some of these outputs and why you might want to do it, I'm going to show you a pangenome accumulation curve. So this is essentially the reverse of what I just showed you, where I removed redundancy. In this case, we're adding uniqueness. So we start with the purple genome. We've got 4 genes there, and they're all unique. So we would have our curve increasing quite rapidly. We've got 4 genes in the pangenome with our first genome. When we add the second genome, i.e. the green one, these first 2 genes, remember, are the same as the purple genome. So we're only adding 2 unique genes. So now this number has increased to 6, and I apologize that this scale is a little bit off. I got it as close as I kind of could. And then by the time that we add our third genome, we're only adding 1 unique gene, so this blue one, and we can see that that curve is flattening out a little bit. So this is called a pangenome accumulation curve. You can have you could have an accessory gene accumulation curve, or pick any kind of subset, but it's mostly done with a pangenome. And what this is really trying to indicate is the diversity and richness of our data sets, and how representative this is of that species or strain or whatever, and how well sampled or sequenced it has been. So we can have what's called an open or closed pangenome, and if this curve keeps increasing, we can say that it's open, and if it was to flatten out even much more than this, we would say that it's closed. And so if it's a closed pangenome, we can say that we're fairly confident that if we sequenced more genomes and added the additional unique genes to the pangenome, that we wouldn't really see much more diversity. We can also then compare the size of the core and accessory genomes relative to each other, or relative to the size of a single genome. In the example that I showed, we had roughly 50% was core and 50% was accessory. And that was just purely for demonstration purposes. This really varies a lot throughout bacterial species. So definitely don't go away from this and think that it's always 50-50. That was just for the example. And then lastly, and I've already kind of touched on this, is that we'll often want to be looking for lineage-specific genes. And again, what we're really looking for in in that case is lineage or strain-specific functions. And again, that might relate back to ecological niches, that might relate back to hosts or disease phenotypes. It might relate to outbreak strains versus endemic strains. There's really a plethora of questions that you can use this type of analysis to answer. So that's the concept of a pen genome covered, and an idea of what you can do with these outputs to answer your questions. But I keep trying to include real elements. So this is actually a list of output files from Rory, one of the commonly used pen genome analysis tools. And I've just highlighted two of the files here. These are two of the files that I've already referred to, the gene presence absence file and the core gene alignment file. There's a list of others here that relate to some of the statistics that I've mentioned, and then you can use to make the gene accumulation curve, for example, and a range of other plots. So how you actually generate this matrix and this alignment. So if we take our pen genome genes, so all of our total unique genes, and we have each of those on a row, and I've colored these here as per the colors I've been using. And then we have across our columns, one column for each genome that we've used in our comparative data set. The matrix is, as I said, literally a binary yes or no of presence versus absence of each of those genes in each of the genomes. So to make the alignment, to make the tree, we first take our core genes. We don't want the accessory genes for these because they're too different, and there are other tools and other analyses that do consider the accessory genome in constructing trees, but I'm not going to talk about that today. We don't want the accessory, so we can essentially discard those. I've discarded them from the matrix up here as well, just to kind of just to show. And so we take each gene and we align them per each genome. So just like the alignment that I showed from BLAST before, instead of having 2 sequences, in this case we'd have 3 of each gene. And then we concatenate those alignments. So basically just string them together and make one master alignment rather than lots of individual alignments. Hopefully you've noticed that the second gene in the green genome is a slightly fainter red-pink to the other ones. And I did this to indicate that although it's the same gene, it has a slightly different sequence. Let's say there's 1 or 2 SNPs. SNPs, again, just to remind you, being single nucleotide polymorphisms. So there's a little bit of variation, but it's the same gene. And what I did this to show you is that how this alignment relates to a phylogenetic tree. So we take our alignment and we plug it into a tree building software. One that I mentioned earlier on is called IQ-Tree. I included the little icon. And so what we see based on the sequences is that we know that the purple and blue ones, based on the core gene sequences, are very highly similar. But because this second gene in this green genome has some SNPs, it would branch distinctly on a slightly longer branch away from the other 2 genomes. So again, this is just to show one of the ways that you can construct a phylogenetic tree and what this might actually kind of mean. We know that this isn't the full story. The tree is only part of what you might want to be looking at and what you might want to communicate. So we can make some more pretty pictures comparing some of the genetic content or metadata of the different strains or the different groups. So I'll go into a little bit about this now, and I will use a real example as well. So we take our alignment, our multi-gene core alignment, And again, just to show you what it actually looks like, it's just some text. It's just some A's, G's, C's and T's. We can see here that there are some dashes. That means that there's a misalignment somewhere. So this is only the first sequence in the alignment. And then so as we go down through the different genomes that we've included, we might have some of the genomes will have bases here where there's gaps. A better way of showing this is in an alignment viewing software. This one's called AliView. And so this much better depicts these relationships than just that simple text file. So you can see that for these, the first few bases, for example, in those first 4 genomes, that they are the same, and then those 4 differ from the bottom 2. And if you look across, you can see that there's little sections of variation, either between, say, the middle 2 genomes and the top 2, or the bottom 2 genomes and the top 4. And so you would get this alignment, these numbers across the top. It spans the entire Pen genome. And so again, we can relate this to a tree. It's a much nicer way of viewing these relationships. And the real life version of this tree would look something like this. And you can see what I've already kind of alluded to with these sequence similarities is that our top 2 genomes are really closely related, and they're a little bit distant from these middle 2 genomes, and they're more closely related by what's called a recent common ancestor to the bottom 2 genomes. But these bottom 2 genomes are even more different from each other compared to the top 2 sets. So these branch lengths and this what's called tree topology is hopefully reminding you of the schematic that I showed earlier. So you've made the tree and you semi-understand what it's showing, at least. What do you do next? So I've been alluding to pretty pictures and visualizations. There's a few things that you can do. I'm not going to talk about this one in pink, hierarchical clustering, but essentially where we had those 3 sets of 2, we might call them different clades. So clade 1, clade 2, clade 3. And there are tools that would do this for you automatically. Using something like that and using these clades and that terminology would really help you make some group-wise comparisons. And again, leading to those pretty pictures and how we might communicate our findings. Because what we're really doing is trying to contextualize our findings with what's previously known in whichever species we're looking at. So again, just kind of heading towards something like what I showed earlier on. And so what this actually would look like, so the final piece of this puzzle is the gene presence-absence matrix. And again, just to remind you, I'm using a schematic. And if we put this against the tree to contextualize the patterns of shared and unique genes or core and accessory genes, I'll add in the real life version. So basically, a lot of blue and white. Blue being for presence of a gene and white being for absence of a gene. And this is being shown in a tool called Fandango, which is a really handy web server. You can drag and drop different outputs, i.e. tree, matrix, metadata, other information, and really nicely interactively do some analysis. So as I mentioned, our blue blocks here represent gene presence and the white gaps are absence. And hopefully what you can see is that we have lots of blue to the left, and there are core genes. And then as soon as we start losing shared genes is our accessory genes. So like my schematic example, we do have here roughly a 50-50 split of core and accessory. And again, I should also mention that these are 6 Yemeni Vibrio cholerae genomes. So again, I am actually using a relevant example. And this is the start. This is one pretty picture that we can make. So we finally got there. Just to expand on this a little bit more, how can you interact with this? What are you actually seeing? How do you know what's what? We can add in we can add the annotations in, and these actually do show up when you drag your gene matrix into Fandango. This is obviously not very helpful, but once we scroll in, we can see a lot more detail. So each of these blue blocks across the top represents a gene, and it lines up with a gene in the dark blue boxes below. If you mouse over these light blue boxes, you get the gene information that we've seen from the Gff file and that we've seen in Artemis before. So this locus tag and product, they should hopefully be somewhat familiar to you now. And this grey bar over here, I forgot to mention, that's our zoom. So that's how we know that we're zoomed in or out and what kind of resolution we can visualize this at. So we've got our pretty picture, and the last thing to add in is some metadata so that we can really start to try and contextualize these patterns. So we've got our tree, we've got our gene presence absence matrix, and then in between here we've got some metadata, and I've just pulled a few pieces of the metadata for these samples. I've included age and sex, as well as location and year of collection. And what I want to really just show here is that based on the relationships that you see in the phylogenetic tree, we don't have, for these 6 genomes at least, we don't have any clustering based on location. However, we do have clustering and similarity based on collection year. So these purple ones were collected in 2018, and they're very closely related to each other, but they're more distinct from samples collected in 2019. And this is also reflected in the presence and absence of the genes that we see over on the right. So lastly, I want to double back on the second pathway. We've gone all the way down the path on the left now, using a de novo and pangenome approach to construct a core gene tree and investigate relationships between these genomes. We've gone from reads to a genome to some pretty pictures. But the other pathway that I mentioned is a reference mapped pathway. This gives us the same end result. We still get a tree, but the sequence that we use is different. And as I touched on earlier, using this pathway relies on some assumptions. The main one, of course, being that we need a reliable, high quality and highly similar reference genome to call variants against in our data set. So I'll talk about this pathway in a bit more detail. So for this example, we take our reference genome, which has 2 genes. It's even smaller than the ones before. And each gene has a sequence as shown here. And we're going to align one set of reads to it. So we've got the reads corresponding to the purple genome. And unlike in de novo assembly earlier, where we align all the reads to each other, we're aligning all of the reads to the reference in this case. Ideally, at each position, you will have reads that overlap with the reference, and you'll also have reads that overlap with each other. And that will give you confidence in the base called at each position. Occasionally you'll have some errors, and if you've got errors, you'll have what's called the consensus sequence. So in this example here, we've got I'm using the black dot as a SNP. It could be a different type of variant, like an indel, but for this example, it's a SNP. But it's only in one of the 3 reads at that position. So that would be an error. Whereas where you have a SNP at, let's say, all of the reads at that particular position, that would be real, and that would be a SNP. So we'd have the purple genome in this example would differ at this base from the reference genome. We'll do the same thing with the green and blue genomes or read sets, and you start to see at different positions we've got some SNPs. The second gene in the green genome is what we would call highly polymorphic. So there's 4 SNPs here relative to the reference, and this is compared to, say, the purple and blue genomes where it's identical to the reference, or even this blue one down here which only has 1 SNP. So once we have added all of our read sets from all of our genomes, we can then work toward our whole genome reference mapped alignment. So this is again in contrast to our core gene alignment that we made using the Pen Genome tool. So where we have the reference map SNP alignment, as it might suggest, we're only taking these polymorphic positions. We're not going to take this first position that had the error. So we can see that the G remains a G, but at the second position that had a high quality SNP, we've got an A instead of a T. And we can do this for each of the genes or across the whole genome, but just using the genes as examples. The green genome at the first gene was identical, so we don't have any SNPs there. And our blue one, we've got a G in place of an A. And likewise, we've got identical second gene in the purple genome. We've got the polymorphic second gene in the green genome, and again, our SNPs are highlighted in bold. And again, identical to the reference is our blue second gene. And so what we do is we just take those polymorphic positions. We don't just take the SNPs themselves. We have to take the equivalent bases from all of the genomes. Otherwise, we end up with essentially a mess, just a lot of bases that don't necessarily go together. But when we know that we're pulling out the same position, then we're properly aligning them. So we take those nucleotides and we align them. So you can see here I've just moved them all up so that they're a bit squished. And then here I've removed all of the excess space. So you can very clearly see the alignment. You can see bases that are the same or conserved between different genomes. So, for example, AA in the purple and green, which is a G in the blue genome, and so on. And this is now what we can use as input for our phylogenetic tree. So I've put here the icon for IQ-Tree, which is a very common tool that we use. And you can see just going again with the schematic, we have a slightly different tree structure from what we got from the core gene tree. Again, reflecting the similarities in the sequence between the purple and blue genome, and then with the green one being slightly more divergent with more differences in the bases from the reference sequence and from the other 2 sequences. So I just wanted to extend this example a little further. If you recall, we actually had 4 genes in our novel genomes, so now I've given our reference genome 4 genes as well. And we've still got these 2 core genes. Our third gene is now this grey gene, and our fourth gene is the yellow gene, which is shared between the green and blue genomes. So if we mapped our reads to these genes, we'd see what I showed before. We've got reads mapping to the core genes. We've got reads just for the green and blue genomes mapping to the yellow gene. So we would get the positions from both red and yellow genes. Again, just whatever the SNP positions were. So if we had a few SNPs, say here and here, we would take those positions for our final alignment. We wouldn't get any positions at all for the grey gene because we don't have any reads mapping to that area. What we would get though using this method that we wouldn't get in a core gene alignment is Enzogenic regions. So you can see here for our green and blue genomes, we do have reads covering the Enzogenic region between the grey and yellow genes. So this could be really useful and important for understanding reasons why genes are lost or missing from certain genomes. And again, just to show that it's really important to choose an appropriate reference sequence, you need something that is closely enough related to your novel genomes that you will get enough informative sites to make a tree. But kind of contrastingly, if you use quite a distance reference, you might get more information in terms of SNPs per gene. Again, it's all relative to the reference. Downstream, you can do pairwise analyses where you can look at SNPs relative to different genomes, not just sort of a singular reference. So again, just a few things to keep in mind, comparing different approaches for different questions. So just to summarize and recap, I've taken you through two fairly generic pathways of genome analysis, which are really dependent on your question and the novelty of your genome. So knowing your questions will really help direct your analysis pathway. So I hope that that's something that you've taken away. And I hope that you've also taken away that there's a lot that you can do with a genome or a few genomes, such as what I've listed on the side and what I went over at the start, particularly for outbreak investigation, comparing across lineages or across different niches. And then you can also look at things like antimicrobial resistance monitoring. One of the other takeaways that I wanted you to get from this is that QC is really important. But overall, I hope that you've now feel a bit more comfortable and have a bit better of an understanding of the concepts underlying genomic and phylogenetic analysis.